So at this point, with the students, what I'm trying to do is show them how scientists figured out what goes on with plants in the air. And initially, scientists were very interested in how plants interacted with light and what that did in terms of the gases in the air. When this work was originally done, we didn't have a good idea about different gases in the air. And during this time, we were just starting to figure out that there were different kinds of gases like oxygen and carbon dioxide that the air was made of. So what I'm trying to do now is set up an environment that will show kids how plants interact with different kinds of gases in the air. To do the experiment, you need a little bit of equipment. The first thing you need is a Ziploc baggie. I usually get a gallon-sized baggie because it's large enough to hold a small plant. You'll need some small plant. It doesn't really matter what kind of plant, although I suggest you get something with bright green leaves. If you can find something like kale that's got bunched up green leaves, that works exceptionally well. Mint plants work well, but any green leaf plant works for this. And what we're going to do is put this plant in a bag, and then we're going to set up an environment of air in this bag that will allow us to show kids what happens with plants and sunlight. So the first part of the experiment involves you kind of sealing the plant in the jar, in the baggie pretty well. I'll leave a little opening because in just a minute I'm going to add some CO2 to the air to make this environment rich with CO2 so students can see what happens to CO2 in the air in the presence of sunlight. In order for me to show these changes, I have to have some way to measure the CO2 concentrations and the oxygen concentrations in the air. There are a number of commercially available products that allow you to do that. If you're familiar with probeware that hooks up to computers, there are CO2 and oxygen probes that allow you to capture the percentages or number of particles of gas in the air. The equipment I'm going to use today is not computer equipment, but it comes from a Japanese company called Gas Tech. Part of the reason why I like this particular equipment over the computer probes is that it provides a static reading. I get a number, one pre, one post reading. When you use the probe, sometimes the figures will jump around and confuse the kids. And I also like this because it's not sensitive to light. When I've tried to do these experiments using the computer probes, sometimes the sunlight causes the readings on the probes to malfunction or to give me error. So what I'm going to use today is a Gas Tech pump. You can find it online. It's sold here in America. If not, you can go to gastech.com and you'll be referred to where you can find them. I'm also going to use two different kinds of detector tubes. I'm going to use oxygen tube, a tube that detects the amount of oxygen in the air, and I'm also going to use a tube that detects the amount of CO2 in the air. Now, to set up the experiment, I want to create this environment inside this baggie that's full of CO2, that's higher of CO2 concentration than's normally in the air. And I also want to create an environment that's lower in oxygen concentration than is typically in the air so that I can demonstrate a change. And to do that, I use this crazy system of putting a straw in here, and all I want to do is breathe several times into the baggie. When you respire, CO2 is emitted from your mouth, your lungs, and I want to fill this baggie with as much CO2 as possible. So just a few breaths. And now I've got a plant it's resting in an environment that's probably higher in CO2 than is normal and probably lower in oxygen than is normal. But I don't want to operate from probables. What I'd like to do is demonstrate for the kids the exact numbers. So what I'm going to do at this point is use my detector tube system, and I'm going to go ahead and measure the amount of oxygen that's present inside my container. The directions for the equipment's online. If you purchase it, it comes with it. But essentially what I've got to do with these tubes is they're sealed tubes. Both ends are sealed so that no air can come in or out of the tubes. And in order to take a measurement, I've got to break both of the ends of this tube and make air go through the tube. To break both ends, I use the little glass tube breaker that they send. I'm not sure what you call it. And I'm just going to break the end of the tube so that there's an opening on both sides of the tube. So now what I've got is a tube with holes on both sides. I'm going to draw air through there, and when the air passes through here, it's going to measure the amount of oxygen present in whatever air I'm sampling. To make this work, I've got to make sure that my arrows point in towards my tube, And now I'm pretty much ready to take a gas measurement from inside here. The only other thing you need to know if you use these tubes is that there's sort of an alignment thing that needs to happen. You've got a red mark and you've got a red arrow. In order to accurately capture the amount of gas, those two things need to be lined up when I draw this arm back so it can draw the air through the tube the way that I'd like to. So what I'm going to do at this point is take my plant in a jar. I'm going to put my plant in a baggie. I'm going to open a very small opening, trying to keep what's inside as much as possible. I'll insert my oxygen tube, and I'm going to draw an air sample from inside of here and see what the oxygen concentration is right now inside my baggie. Now you may have heard a click. That click tells me that the sampling is complete, but I want to leave the tube in here for about 30 seconds to make sure that I have an accurate reading. So here we go. I can see some change happening, so let's go ahead and take a look at what our measurement is. To determine what our measurement is, you need to look at the scale that's on the side of the glass tube, and if you look, you'll notice some numbers starting at 8. There's a 9, there's a 10, there's a 12. So if I follow up to where the white lines end, that tells me about how much oxygen is inside this baggie in terms of the oxygen in the air. And right now, it looks like there's about 18 or 19% of oxygen in the air inside this bag. That's a little less than is typically in the air. Usually in the air, you have about 21% oxygen in the air, in a healthy air. So we do have an environment where there's lower amounts of oxygen than we typically would find outside. Now let's take a look at the CO2 in the jar and see what kind of levels, in the baggie, and see what kind of CO2 levels we have in the baggie. And to do that, I've got a second tube that's a little smaller. This measures CO2 concentrations, and I'm going to play the same game. It's sealed on both ends, so I need to break that open. Make sure that I've got both ends broken. I look for my arrow. In this case, it's an orange arrow that has to be pointed in towards my detection tube device. And now I'm ready to measure the CO2 concentration inside this, the air inside the plastic bag. So one more time, I've got to make sure that my red arrow and my red line up. I'm going to play the same game. I'm going to just break open the very small corner of this baggie, stick it in here, seal it up tight. So now I've got to draw the air sample out of the container. Okay, again, you probably heard the click. I want to wait a few minutes, and if the video is picking this up, you may notice that there's a lot of purple forming on this one, which is an indication of CO2. So the more of that purple, the more CO2 there is. So we're getting a high amount of CO2. I'll leave it for just a few seconds to make sure I'm capturing all the accurate measurement that I can. And let's take a look at our CO2 measurement before we do the next part of the experiment. If you look, this particular tube measures the concentrations from 0.03 to above 1% of CO2. So right now, there's more than 1% CO2 inside this container, which is a high amount of CO2. Typically in the air, the amount of CO2 is less than 10%. In the air, it's usually 0.03%. So less than 1% of CO2 is usually in the air, and we've created an environment that's pushed that amount of CO2 to above 1%. So right now, this plant's trying to survive in an environment that's got low oxygen and high CO2. And now what we're going to do is we're going to take this setup, we're going to take it outside, and we're going to put it in the sun. And we want to give it a chance to do photosynthesis. Photosynthesis takes about 10 minutes to start. So if you want to get a good reading, you'll need to allow for at least 30 minutes of time. You'll want to put the baggie in a place where the tops of the leaves get a lot of bright sunlight. Give it 10 minutes to start photosynthesis, and then at least about 20 minutes to do some work so you can see a change with kids. So what we're going to do now is take our setup outside, find a sunny location, sit it there for 30 minutes, and then we're going to take a post reading of the CO2 and the oxygen concentrations to see if there's been any change in these levels of gases in the air because the plant sat in the sunlight. So all you need to do is find an outside spot, take your Ziploc baggie, keep it sealed, place it in the sun where it's going to get lots of sunlight, particularly the tops of the leaves, and leave it for 30 minutes. We'll come back in 30 minutes and take a post measurement and see what happens. So we've taken our plant outside. It's been in the sun for 30 minutes. Now we've returned and I'd like to take some post readings of the CO2 and oxygen concentrations. Just like before, we're going to break the ends of our detector tube. We're going to make sure that the arrows face into our detection device. I'm going to line things up. I will just briefly or just barely open this container to allow very little to come in or out. And I'll take a sample of the oxygen concentration or percentage that's now inside our sealed container that's been sitting in the sun for 30 minutes. I'll let it sit for a minute to make sure that it does what it needs to. And now when I take it out, it's kind of hard to see, but what I'd like to do is compare this post reading to the pre-reading. Okay, so if I can get a little, I've got my post and I've got my pre, and if I line those up, and we can see that now after sitting in the sun for about 30 minutes, our plant has restored the amount of oxygen in that sealed environment to around 21%. In fact, it looks a little higher than 21%. So now our plant in the jar, our plant that's been sealed, has increased the amount of oxygen in its sealed environment from around 18% to over 21%, which is normal. So now let's see what's happened with the CO2 concentration in our container. I'll play the same game, break both ends. Take my tube, make sure that the arrow faces in, line my arrows up, and let's take a reading of the amount of CO2 that's present in our sealed environment. Now as we wait, you'll see the purple, if you can, I'm sure if the camera can pick this up, there's a little reaction starting to take place. The purple line's starting to grow a little. We'll let it sit for just a few seconds to make sure that we're getting as accurate a reading as we possibly can. Now let's see what's going on with the CO2. Okay, again, this is the post reading. This is the pre-reading. If I put the two together, let's see if I got these right. And if you look at the scale, I'm turning these around on you, you'll notice that now in the camera angle, the pre is on the right, the post is on the left, and I position these so that you could actually see the numbers. You'll notice that the amount of CO2 has decreased significantly inside this sealed container. And what I'm trying to demonstrate, just to be clear, is that the plant has increased the amount of oxygen in the sealed environment and it's decreased the amount of CO2 in the environment. This is important for helping kids begin to conceptualize photosynthesis. So one of the things I usually do here is begin to construct a word formula that puts together the different pieces of the formula as I learned it. The one piece that's missing in this series of activities is the starch piece. One of the things that scientists figured out was that if you put a plant in sunlight, that the leaves in sunlight help produce starch in plants. And so I'm going to give you a URL that you can go to and find an activity that shows you how to measure starch in plants. I usually accompany these activities with that particular activity, which is a well-known common activity done in many middle and high school science labs, and use the combination of these different experiences to help build a formula for photosynthesis that the kids can actually see. It's not asking them to imagine what might be happening, but it's actually showing them how plants use CO2 and make oxygen and make starch. And so at that point, I'm going to ask you to go back and look at the models that you created to begin with and revise your existing models to include this new information that you may or may not be new information about what plants do in the presence of sunlight. I'd also like you to consider any new questions that this information at this point raised for you.